So hi everyone. First of all, I would like to thank the organizer for the invitation to be here. And I will start with a confession. So this is a very different audience from what I'm used to. So I'm not used to talking to a room full of bacteria and yeast microbiologists. So I come from so we make vaccines and we study viruses. But so whatever questions you guys might have today about yeast and the expression system that we are working with, there is a room full of people who I can enlist to answer those questions. So that gives me a little bit of comfort. So that's that. So now we're gonna start. I will stop using PDF from now on because last time I gave a talk, it takes like 10 minutes. Okay. So should I ต้องไปตรงขยับเอาไหมคะ Okay, so let's set the stage here. So we've been hearing wonderful talks about, but maybe not wonderful, climate changes and how the world is basically burning down and how we all as scientists will have to fight that with, degradation of plastics and making alternative food and stuff like that. So my contribution to that would be keeping steady supply of bacons until we get to that point where we have alternative food. So this is what swine farming is in Thailand right now. So we have swine farming with very different types of farms. We have like really big farm, very well managed. All the way to like basically backyard small farmers, and the number of farmers where we get from the DLD, which is the Department of Livestock Development in Thailand, has decreased quite a bit. So in 2018, it was about 184,000 farmers, and in 2023, there are 150,000 farmers. And most of the people who are missing is basically small farmers who couldn't cope with the large outbreak of African swine fever, which hit very hard during around like 2018, 19, 20 to 22 in Thailand, the whole Asia in China, Vietnam, Cambodia, Laos, and Thailand. So most of the people who fold and had to like to do the total depopulation of their farms are small farmers. So this is the breakdown of how farming in Thailand is. So basically a large proportion of farmers, 90% of the farmers basically have like 50 pigs to work with, right? So those are the people who we really care about the most. They're like, don't have too many tools and don't have enough fun to do really good biosecurity so that they can fight off the outbreaks. And these are the challenges in Thai and Southeast Asian swine farms. We face it almost constantly. So you can see that in here, it's a PEDV, it's a type of virus that cause diarrhea. And a really hard hit PEDV outbreak can basically take away about 2 to 3 million piglets out of the system a year. And we have a constant threat of PECV2, porcine circovirus 2 epidemics. And you can see that the disease can make the pigs much smaller than what the normal pigs would be. And this is ASF. So ASF, African swine fever, basically just kills the pig, almost like 90 to 100% mortality. And for us who are not small farmers, like normal consumers of bacons and pigs, would feel this kind of impact in the price, right? So for ASF, we had like about 40% jump in pork price within like a few weeks time. So that's what the problems is in Thailand. So now, maybe like 5-6 years ago, we thought that one of the reasons, one of the ways to take care of the problem is that we have to have some kind of a regional capacity to make or manufacture vaccines or veterinary therapeutics in Thailand, because most of the vaccines that are being used in farms right now are imported, and that is expensive. So it caused a lot of gaps between big conglomerate contract farms and the small farmers, who are like 90% of the farmers in Thailand. So and another reason was that to increase food security in the region. And there's also a scientific reason for this, because pathogens that are circulating in Thailand would look different from whatever that's circulating in Europe or in America, where the vaccines are produced and done research with. So in 2017, with the leadership of Professor Colin Robinson here at the University of Kent, we teamed up and got a funding from GCRF for the project called, basically it's the capacity building project for manufacturing biopharmaceutical and animal vaccines in Thailand and in Southeast Asia region. This is the group from, there are hearts in there. Okay. So this is the group at the beginning of the project, and we took this picture at the NBF, which is the National Biopharmaceutical Facility at King Mongkut Technology University in Thonburi. Right. The project aims to build capacity in manufacture recombinant protein-based biopharmaceutical molecules and veterinary therapeutics. So this is basically the pipeline of how the research in making vaccines work, right? So you first have to design what antigen you want to make, then you choose what kind of platform you want to make your antigens with, and then we do some process development. We have QC and then translation to the end user. And at each step of the way, we have teamed up with people who are an expert in that field. Right. So for the upstream process, we have University of Kent and Biotech. And then for the downstream process, we will hand it off to UCL and Ken UTT, who owns the NBF facility that I mentioned. And then we have people from Imperial College who helped us do the mass spec and quality control of the molecules. All right, so today I'm going to talk to you about two molecules that we used as models for this project. The first one is PCV2D subunit vaccine. It's a vaccine against a small virus. And then the other one is porcine interferon alpha, which is basically a biomolecule to be used as an immune modulator or a drug for pigs. So porcine circovirus is a small virus, and it basically has very few proteins, and the protein that is immunogenic is basically the protein that makes the shell of the virus, and it's called the capsid protein. So the antigen will have to come from the capsid. So when we try to choose the production platform, we were doing it in 3 different places. So University of Kent will take on the E. coli platform, and the UCL was going to work with Pierre Pasteuris. And at Biotech, we had basically, the team here at Biotech and TBRC developed the Hansanula lookalike platform for recombinant protein production. So we took that and then we said, okay, we're gonna try that also. The PCV2. So I'm gonna show you from the OT yeast first, which is the Ogatia yeast, and the expression vector was also developed in-house in biotech. We basically just took the PCV2D sequence, which is the sequence that we got from the surveillance of the farms in Thailand between 2011 and 2015. And then we do the codon optimize and try to express it and secrete it in the OT yeast. And after a lot of optimization, we could produce it here. But our best final yield was only about 120 micrograms per milliliter of culture, which is quite low compared to what Colin's group at Kent has seen working with E. coli. So for E. coli platform, with the same construct, they could make about 1 grams per liter culture of the protein. And so then because of the yield that's much different, so we took the E. coli platform and then do the downstream process in 2 independent places, which is at UCL and at KMUTT. They both independently did 20-liter fermentation and could do this one-step purification process, just like what the small scale people have done. And they got the yield to about the same, which is 1 grams per liter of culture in the large scale fermentation. And then we were the group that test the antigenicity of the antigen. So we tested in animals, and we found that both the antigens from OT, which is the yeast, and the proteins that were produced in E. coli were antigenic in animals. And it can induce neutralizing antibody inside the animals so that they can be used as a vaccine. So that was the first story. We made the vaccine, and we started out in 3 different platforms and then the bacteria one. Now, I'm going to tell you about the second story where it's now the yeast turn to win. All right. So we started out the same way. We used 3 different platforms. And Colin's group at Kent, people were trying to express this molecule, which is the interferon protein in E. coli. I think they tried for like a year or a year and a half and tried different constructs, and they couldn't really get it to express. But at the same time, we used the similar yeast system that we used with the PCV2 vaccine, and we did get it to express. All right, so just the background, porcine interferon. So interferon is basically a biomolecule that is induced when you all are infected by mostly viruses or other pathogens. It basically is a molecule that is sent out from the infected cells to tell the neighboring cells that, hey, be prepared, you are going to find some pathogens coming. So it mounts enough response to negate the viral replication and the disease. Right. So what we've seen in the past in livestock work is that the molecule, because it is from the innate immune innate immune response. So it's non-specific to the type of pathogen. So it's unlike the antibody when you are immune because you have taken the vaccine for COVID, then you mount an antibody specific to COVID. Interferon is different. So it's a molecule that's made from viral infection. So it will most of the time negate any virus. So in pigs, there have been research even in animal trials looking at the antiviral activity of the porcine interferon, and it's been shown that it can show slight reduction in African swine fever viral load inside the pigs. For other very well-known pig diseases, PRRS virus, classical swine fever, and foot and mouth disease virus, also they've shown that pigs, when received the treatment of porcine interferon before or at the same time of viral infection, can basically showed less clinical signs. And interferon is a well-known human drug, right? So people with hepatitis C viral infection, chronic infection, or some kind of cancer will receive interferon treatment. In companion pets also, when when a cat is infected with chronic feline virus, the veterinarian will treat it with the feline interferon omega also. So it's not very strange idea to try to treat animal with this biomolecule. The problem is, to use it in livestock, you have to beat the price. So you have to minimize the cost of production. So that was our idea of using the biotech own recombinant protein production system to try to beat the cost and see how far we can go. So this is where I will not be very good at explaining all these things, but what I want to say is that we have a lot of different hosts, and we have a lot of different expression vectors that differ by mostly the promoter and the signal sequence. Yeah. So we try combinations of these, all right? So we picked the OP39, which is Ocatia polymorpha, which is the 39 came from some numbers that TBRC gave it. And this is basically a Thai strain of Hansenula polymorpha. And we try different expression vector. Okay, so this one is the one that we wanted it to work, but it didn't. So this one has a maltase promoter, which is it can be induced. The expression can be induced by sugar, table sugar. So it would really bring down the cost, and then we don't have to use methanol as an inducer and whatnot. But we didn't get any expression of our protein from this combination, so that's out. We also try the MOX promoter and FMD promoter, which are the methanol inducible promoters that are indigenous to the OP39 strain. And we found that the expression level that we get from these two indigenous promoters is actually lower than when we use the heterologous promoter from a different Ocatia strain. So I think in the end, we stick with this construct of vector. All right, so in order to try different combinations to get the best, the highest expression level, we try to make 2 copies of the expression cassette into the plasmid and then integrate the plasmid into the host via cloning. But we saw that even when we have two cassettes inside the host cell, we still didn't get as high level of expression when compared to when we use the construct of POT switch, which is the promoter that comes from a heterologous Ogatia strain. So and we did we screened many clones. All right. So in the end, I think we took this pair, the host that is OP39, and the expression vector that contains the methanol inducible promoter that comes from a heterologous Ogatia strain. Right. And then we could show that our construct, or the recombinant clone that we picked, is expressing the protein porcine interferon alpha as well as the commercial Pichia pastoris system. All right, and then we got a nice exchange grant from IVBN, which is funded by part of GCRF, and we did the first fermentation experiments using the benchtop fermentation system at Kent, with the help of Alicia's lab also, and Connor here. So this is our research assistant spending, I think, 4 weeks at Kent trying to express and secrete this thing into the media. And that was the first time that we realized that changing from a shake flask culture where we routinely and normally get the proteins to the level that we want into us 4 weeks of trying to find the protein. So we spend a lot of time with changing a lot of fermentation condition. And I think finally in the last week, we got to see this tiny bands of the protein from the fermenter. And so that was the first sign of, hardship along this journey. We do work, we came back and we do work with our collaborator in biotech, who is an expert in doing yeast fermentation. So this is a lot of work also that I would be really bad at explaining. So they did a lot of fermentations and I think changed a lot of conditions, changed a lot of buffers, changed a lot of numbers of hours to grow the yeast inside. And I think at this point, we are just catching up with whatever we have in the shake flask. So we used to work from no bands at all to now we routinely see the bands in the fermenter. However, that's a lot of work. And if I present this to the person who gives me money, they would still not be happy. So at this point, we're still working towards the goal of 2 grams per liter. But we realize now, I mean, the goal was set when we were doing shake flasks for like about 0.7 grams per liter. So that didn't seem too bad at the beginning. But now I think after a year of trying the fermentation and going from 0 to 0.7 grams per liter, we think that is still going to take us a while. However, we took the protein, the recombinant protein that we got from the fermentation or from this yeast that's produced from this yeast, and we could show that it has a normal biological activity. So against PRRS virus, this is the virus that basically cause baby piglets to die. Not baby piglets. It cause miscarriage, miscarriage of the sows, this virus. This virus is the ASF, which is African swine fever that caused the pork price to jump 40% in 2 weeks. And this is the PEDV virus that caused diarrhea in piglets and have a mortality rate for piglets of about 90 to 100%. And we can show that all these viruses are sensitive to the interferon that we made. So this is what normal cells look like. This is the cells when they are infected by virus. So basically these viruses kill the cells and the cells detach, and then you would see these like lighter circular blobs, which look like this. This one is easier because we made the virus so that the virus expressed the red fluorescent protein. So if you have a lot of viral replication, you see a lot of red fields. But if we treat these cells with porcine interferon before the infection of the virus, we can see that the cells look almost like the normal cells, just like when there's no virus. This one also has very, very few blotches of red. So this is working. All right. And I can't show you because it's not very legal too, but I'm just going to mention. So we do have our collaborator in BIS group, which is one of the veterinary distributors in Thailand, and they work a lot with animal farms, and they have basically veterinarians consultants at each farm, and they did a small limited field trials using the interferon that we made. And for the limited small trials, in the midst of ASF outbreak, we could show that without interferon, you would have to kill the word they use is culling. So you would have to cull a lot of pigs, and they would have the loss of about 46%. But if they use the interferon before they move the piglets into the pen, they could reduce the loss down to about 5%. So what we are trying to do now is trying to make this work in a fermenter in a larger scale production, and try to get money to do the animal experiments. All right, with that, I thank the lab, the virology and cell technology lab at Biotech, and everyone who's contributed to the project, and the funding from UKRI, GCRF, and NASDA, and PMUC. And especially the collaborators. We have wonderful collaborators from England in the GCIF projects, and also our friends in the yeast group at Biotech. I'm so sorry, I don't remember the lab's names. IMCT, right? Yes, Microbial Cell Factory lab, especially Dr. Niran, who's not here, who's been helping us with all the yeast work. All right, with that, I thank you for your attention, and I would love any comments or questions that I can distribute to other people to answer.
Thank you, Dr. P, for enlightening presentation. So we have only 1 minute for one questions.
Thank you for sharing a wonderful story and like impressive story, like how to get interferon. So like, I just think that, okay, you described that you improve the yield by scale up like the conversion from flask to fermenter, right? So like I think it is possible to improve the solubility of interferon itself by adding like some tag, like flag tag or the sumo tag that improve the solubility of the protein in host cell. Like
You
I
mean
know,
in,
like for some protein, like you didn't express well, but you add like some tag, like sumo tag, or making a fusion protein, it can improve like a tenfold expression.
yeah. So you mean in bacteria or in yeast? In yeast.
I know that in bacteria it's work well, but
Yeah.
for in yeast, there might be like some tag that might improve the solubility and the like expressibility of the protein.
Yes. Okay. So, yes, we can definitely try that. We have not tried that. We can try that. But I think what we thought at the beginning was that we are trying to make it because it will in the end have to be a drug. So so people try not to put tags on it or not to put to increase the steps that it has to go through during the downstream process because that will basically increase the production cost. And I think in the beginning we were aiming for that. So that's why we didn't really, we did a lot of screening instead of trying to put the tags on or to fiddle with the construct itself. Yes. But thank you.